In this video, follow along step by step as we explore how the leading and lagging strands can be simultaneously synthesized by the 3 DNA polymerases found in the replisome. Each DNA polymerase is tethered to the replisome via a tau subunit. We can label these DNA polymerases with numbers 1, 2, and 3. At any given moment, only 2 sliding clamps are attached to the DNA, while the third remains associated with the clamp loader within the replisome. It's important to note that the DNA polymerases, and not the clamps, bind directly to the tau subunits. In step 1, DNA polymerase 1 is attached to a sliding clamp and engaged in continuous synthesis of the leading strand. This role remains constant throughout the process. DNA polymerase 2 is also associated with a sliding clamp and is bound to a primer that was previously synthesized. It will be responsible for the synthesis of an Okazaki fragment of the lagging strand. DNA polymerase 3, while still bound to the replisome, is inactive and not associated with DNA. In step 2, DNA polymerase 2 continues synthesizing the current Okazaki fragment on the lagging strand. During this step, the replication forks move to the right and expose some of the lagging strand templates where a new Okazaki fragment can be synthesized. During this time, primase synthesizes a new RNA primer to initiate the next round of lagging strand synthesis. In step 3, DNA polymerase 2 continues elongating the existing Okazaki fragment, while DNA polymerase 3 remains inactive and bound to the replisome. Meanwhile, primase completes the synthesis of the new RNA primer and then dissociates from the DNA. The clamp loader complex attaches a sliding clamp to the newly synthesized primer. In step 4, DNA polymerase 2 completes the synthesis of its Okazaki fragment. DNA polymerase 3 binds to the sliding clamp that is attached to the new primer and starts synthesizing a new Okazaki fragment. In step 5, DNA polymerase 2 completes the synthesis of the Okazaki fragment and dissociates from its DNA template and the sliding clamp. However, DNA polymerase 2 will remain bound to the replisome and inactive until it begins the synthesis of a new Okazaki fragment. DNA polymerase 3 starts the synthesis of the next Okazaki fragment. In conclusion, DNA replication is the result of the coordinated action of 3 DNA polymerases and is controlled by a large protein complex called the replisome. The replisome is responsible for the simultaneous synthesis of both the leading and lagging strands. The replisome contains 2 active DNA polymerases that synthesize the leading and lagging strands, and 1 inactive DNA polymerase which is ready to be loaded onto a new primer and elongate the next Okazaki fragment. The structure and function of the replisome allow for the simultaneous synthesis of 2 DNA strands in opposite directions.